Stereoisomerism, cis-trans, and EZ isomerism. So far we've talked about structural isomerism. This is like when we break up a carbon chain. Those are structural isomers of one another. If we move around a halogen, 1-chloropropane, 2-chloropropane, those are structural isomers. Or if we mix up functional groups like propanone and propanal. So those are all examples of structural isomerism. So you have the same molecular formula, the same number of each element, atoms of each element in the molecule, but the atoms are bonded to different atoms in some way. Stereoisomerism is different because all of the atoms are still bonded to the same atoms, but they are oriented differently in space. At first that sounds really odd, but hopefully by the end of the video that will make more sense how that's possible. So it's all about orientation. Now in stereoisomerism we can actually break that down further into conformational isomerism and configurational isomerism. This term we're going to go over for just a second. It's kind of a minor add-on in the syllabus. You should be aware of it. I don't really think you need to be aware of configurational isomerism as a term, but we are going to look more in depth at two kinds of isomerism that fall under that category. So a conformational isomer, really just to be aware of the term, happens when we have a single bond, and the single bond can rotate. And as a result, when the single bond rotates, you can end up with slightly different orientations of groups sticking off of the atoms. So if you look here, you have a molecule of ethane, C2H6. And so normally when we draw it, we kind of simplify it. We draw all these bonds like right angles, even though each carbon atom is tetrahedral sp3 hybridized, and so we know that the bond angles are actually 109.5. And so this drawing here, which uses the dash wedge type of structure to try to indicate the three-dimensional nature of the structure, gives a bit better picture of what it actually looks like. And then these things down here called Newman projections, which really are not a part of the IB syllabus, try to show you what it would look like from end on. So if we were looking at the molecule from one of these sides, straight down the bond axis between the two carbons. And you can see that you could take those hydrogen atoms that are on each carbon, and they could be staggered in how they're oriented, or they could be lined up right with one another, which is called eclipsed. And so there's different orientations that those attached atoms can take around the carbons, simply because the bond can spin and the bond can rotate. So single bonds are able to rotate, therefore you can end up with these things, which are called conformational isomers. Now, as far as I can tell, all you really need to know about for the DP syllabus is that conformational isomers exist, and it has to do with different orientations from bond rotation. Now, I think it's important to cover that first because this kind of isomerism, which is far more important for the DP exam, is about isomers that occur when you have bonds that cannot rotate, so restricted rotation. Now, a bond can't rotate really, and there are two kinds of examples where the bond can't rotate. There are C to C double bonds, or really any double bond, because the pi bond that we have around the two atoms, remember it's above and below, if we try to spin that bond the way that we just rotated that single bond above, it would cause that pi bond to break. So alkenes, in particular for this topic, have that pi bond, and therefore the bond can't rotate, and so we can end up with different orientations locked into molecules. And so the classic way to name these is cis and trans isomers. When you have this bond that can't rotate, so these are both 2-butene or but2ene. In this example, you can see that there are methyl groups sticking off of the carbons in the double bond, and in this one they're both oriented in the same direction, whereas in this one they're oriented in the opposite direction. And so that's really all there is to this naming system. and to the difference in these types of isomers. Cis meaning same, and trans meaning across. So on opposite sides. This also occurs in cycloalkanes, which we've mentioned before can be structural isomers of alkenes. They have the same number of carbons and hydrogens, C3H6. For propane, same general formula of CnH2n. When they form the ring, that also means that the bond can't rotate, because the ring would break if we rotate the bond. So now I have down here a cyclohexane and another cyclohexane. It's dimethyl cyclohexane, and those two methyl groups can end up pointed in the same direction, and we'd call it cis, and these ones point up and down, and we'd call it trans. Now, you don't have to name cycloalkanes for the DP syllabus, but if they presented you with a structure, you should be able to identify if an isomer is cis or trans. Okay, so we're looking for the same attachment, in this case a methyl group, and is it oriented in the same direction or in opposite directions, and that's really all there is to it. Now, it's important to note also that not every alkene or cycloalkane has cis-trans isomerism. Some of them just don't. For example, but-1-ene. If you look at this one, we can think about flipping the groups that stick off of the carbons, and it wouldn't make any difference. So if I flip these hydrogens, it wouldn't make a new molecule. It would just be the exact same molecule. And if I think about flipping these two groups, and the ethyl group comes down here where the H is, and the H goes up where the ethyl group is, I would really have the same molecule. If I flipped the whole thing 180 degrees, it would still look the same. It would be the exact same molecule. So not all molecules can have cis-trans isomerism. So it's only when we have carbon-carbon double bonds or cycloalkanes, and then we have when we have two identical attachments that are either pointed down in the same orientation across the double bond, or in opposite orientations across the double bond or the ring. And there's some opportunity for practice here. Now, just like not every molecule has cis-trans isomerism, not all isomers that exist in this way can be described very well by the terms cis and trans. It's kind of a limited system, because really, as you saw in the previous examples, it really has to be two of the same thing across the double bond or across two atoms in a ring, like two methyl groups or two chloros. But what do you do when you have different atoms? So in this example, F, Cl, Br, and H. So I've got 4 different attachments across this double bond. And now, if you look over here, I've made a different isomer. I've flipped the F and the Cl are oriented differently in the 2 different molecules. So these are isomers of one another. They are stereoisomers of one another. But how would I describe them in cis-trans? They're all different. It's hard to tell you what's on the same side and what's across from each other. So in this system, we have to assign priority to the atoms. And it's really simple. Higher priority goes to higher atomic number. So of the 2, the chlorine is a higher priority, the fluorine is a lower priority on that left carbon atom. And on the right, the Br is higher priority and the H is a lower priority. So in this example, the 2 higher priority things are on the same side. And in the next example, the chlorine and the bromine, the high priority things, are across from each other. So what are we going to call them? Well, it's no longer cis-trans. We now call it E and Z. E, well, the way to remember it is treat the things like enemies. It's not actually where it comes from. The terms are German. And Z, remember that they're on Z, Zame, Zide. They're on the same side. So a Z isomer, the higher priority atoms or groups are sticking off of the same side, kind of like cis of the double bond or the ring. And in the E system, they are enemies from each other, and the higher priority groups are across from each other. And that's where we're going to pause our video.